A cDNA library is a collection of cloned DNA sequences that are complementary to the mRNA that was extracted from an organism or tissue. During the first step of preparation of a cDNA library, the total mRNA from the cell type or tissue of interest is isolated. These mRNAs have 3' poly A tails. Due to their 3' poly A tails, mRNAs are easily separated from the rRNAs and tRNAs present in the cell extract. In the next step, the oligo-DT primers or thiamidylate primers undergo hybridization with mRNAs. As a result, oligo-DT primer mRNA hybrid molecules are formed. In the next step, the reverse transcriptases transcribe mRNAs into cDNAs. As a result, cDNA mRNA hybrid molecules are formed. cDNA stands for complementary DNA. In cDNA mRNA hybrid molecule, this is the mRNA and this is the cDNA. In the next step, the mRNAs in the cDNA mRNA hybrid molecules are removed hydrolytically with the use of alkali, and 3' poly tails are also added to the cDNAs. As a result, a single-stranded cDNA is formed. In the next step, oligo-DC primers undergo hybridization with previously formed single-stranded cDNA. As a result, oligo-DC primer single-stranded cDNA hybrid molecules are formed. In the next step, a complementary strand is synthesized by using single-stranded cDNA as template in presence of DNA polymerase. As a result, double-stranded cDNAs are formed. In the next step, the EcoR1 sites in the double-stranded cDNAs are methylated. As a result, methylated double-stranded cDNAs are formed. This methylation protects the cDNA from subsequent restriction enzyme cleavage. In the next step, the EcoR1 linkers are ligated to the double-stranded cDNAs by using DNA ligases. These DNA ligases are extracted from bacteriophage T4. The EcoR1 linkers are restriction site linkers. As a result, the EcoR1 linker ligated double-stranded cDNAs are formed. In the next step, the EcoR1 linker ligated double-stranded cDNAs are cleaved with EcoR1. The EcoR1 is a restriction endonuclease. The EcoR1 restriction endonuclease specifically cleaves the attached EcoR1 linkers. As a result, one sticky end is formed at one end of each strand in the EcoR1 linker ligated double-stranded cDNA. In a separate procedure, the plasmid vector is cut with EcoR1 restriction endonuclease. As a result, plasmid vector having appropriate sticky ends are formed. The AMPR sequence in the plasmid vector acts as transcriptional activator. It is a selectable marker. The plasmid vector becomes more useful for the cell in presence of a selectable marker. In the next step, in presence of DNA ligase, the previously formed double-stranded cDNAs with sticky ends ligate to plasmid having appropriate sticky ends. As a result, DNA molecules are formed. These DNA molecules are transformed into E. coli cells and select for MPR. As a result, individual clones are formed. Each clone carries a cDNA derived from one mRNA. Now, advantages of cDNA library. A cDNA library only contains the DNA sequences of active genes in the cells or tissue. A cDNA library has cloned genes containing only exons, means a cDNA library has cloned genes with no introns. As a result, the length of the eukaryotic gene is decreased. This shortened length of eukaryotic gene makes it more feasible for recombinant DNA manipulation. As there is no introns in the eukaryotic cloned genes of a cDNA library, the bacteria can synthesize the correct proteins from the eukaryotic cloned genes of the cDNA library. Please like, subscribe, and share.